All right. Hi, everyone. Welcome back. This week is we're going to talk about turfgrass biology. So we started with a little bit of the history in the background. I thought it'd be a little fun to get into that aspect of it and just how turfgrass became so prevalent in our landscapes today, or how I feel that they did, connecting the dots. And now we're going to get into the biology. We talked about some of the value of it in the landscapes, and now we're going to get into the nitty gritty of turfgrass. So here's some basic terms. I don't know what level of science, biology, chemistry, so on and so forth everybody has. So I guess I'm going to try and take something complex and hopefully I'm going to do a decent job of boiling it down to make it simple for you guys. Again, not knowing what everybody's background or level of interest in the science aspects of this stuff is. That's my entire life. As I've said in the past, I started off actually going R&D, and I was going to go more of like either the R&D route or a tenure track, which I'm kind of doing now. Anyways, that was my background coming into it. I actually worked pretty extensively with the genetics side of it. Complicated if you're going into a molecular biology route, and that's what I actually started off in. So it was really cool, and something that I was actually really interested in possibly going that route through school, but. I actually also enjoy helping people. And one of the things that I learned through my work experience over the last 20 plus years is that I actually enjoy helping people and using my knowledge to help people. And one of the things that I really kind of depressed me being in the research and development aspect of it is, I mean, you're doing all of this great work, but what value does it have? And yeah, at the end of the day, when you get done with all the nitty gritty of what you're doing, you're hopefully potentially going to bring a product to market that is going to help people. But one of the aspects of one of my early jobs when I worked for Scotts and Marysville is I would actually go into Home Depots on the weekend. There was a Home Depot on the north side of Columbus that I would go to on the weekends. So every Saturday and Sunday through about a 2.5 month period in the spring and about a 6 week period in the fall, I would actually go into the garden center at Home Depot and I would stand out in the fertilizer aisle, and I would have all my Scotts logo and apparel on and everything. And I was there to actually consult with customers that would come into Home Depot and help guide their product decisions and help them troubleshoot. And I got questions all over the place. So a lot of stuff that didn't necessarily pertain to fertilizer and lawn management in general. I mean, I would get a lot of questions from a lot of different aspects. So I really enjoyed that, and I really enjoyed helping people problem solve. And so I guess if I hadn't gone into this, maybe I would have been a doctor, because I really enjoy helping people and I love doing kind of investigative forensic type stuff. So I guess that's why plant pathology really appealed to me, because you're kind of walking into a scenario without much information, and it's kind of a little bit of being a doctor. It's a little bit of CSI. It's a little bit of everything. You arrive and there's a dead body on the property. The only benefit of it is really that, next to medicine, my patients, if they do die, if they do succumb, I'm probably not going to get sued, and nobody's really going to care, and we can always go in and fix it. So, my patients don't usually talk back, and if they die, I'm probably not going to get in trouble for it. So, a little bit safer, I think, than going into medicine. So, let's get into the biology aspect of it now, and some basic terms when we get into to biology. If you've had biology in the past, you've probably heard these before. So we're going to start off with taxonomy. This is a system of classification. It used to be based on visible traits. So when you look at a plant, especially, and some of you, I believe a requirement was plant ID before this, so I'm sure you're probably familiar with this term. It's a system of classification. So back, if we look back to like the Orto Botanico in Italy and people like Linnaeus and people that were really getting interested and involved during the Renaissance period in plant identification, classification, nomenclature, all of that. They recognize the importance of being able to group and identify plants. And this was based on similar visible traits. Today, in the 21st century, now that we're in essence in the crypto era, we rely on genetics. So we can actually genetically map these plants, and that's how we do things now. So genotype. So this would be the combination of genes that define an organism. So you have a genotype, I have a genotype. Our genotypes share some similarities because if you look at all of us as human beings, we are very similar in a lot of ways. Overall, like we usually have a head and two eyes and two ears and a nose and a mouth, arms, legs, feet, so on and so forth. But each of us is uniquely different from one another. And so the genotype that makes us up overall, all that commonality, that's the collection of genes that define an organism. How you and I appear differently is a visible expression of that genotype in the form of identifiable traits. So do you have brown eyes? Do you have green eyes, blue eyes? Do you have blonde hair, brown hair, black hair, skin color, your height, your weight? A lot of these things are determined by your genes, and actually specifically what genes are turned on and off. So, and this is really important to the genetic aspect of it when we get back to taxonomy. So as we transition into this information era and the genetics and being able to genetically sequence and profile organisms, we can better determine if they really are genetically distinct, or if possibly some of these variations that we see are phenotype related, meaning that the specific genes are turned on and off. So sometimes phenotype is going to be a representation of a completely different genotype. Sometimes a phenotype is going to be a representation of a set of genes and then more of a determination. Like that a human being, for example, can have any number of hair colors, any number of eye colors. So, that phenotype is going to be a set of traits that can be seen. And so if you think about it in the plant world, that would be more what we talk about with cultivar selection. So when you have a specific variety, okay, so this would be a variety or a cultivar of a type of plant, means that genetically, genotype or genotypically and phenotypically, that plant shares the same genetic characteristics or the same pool of genetic characteristics, but a cultivar variety is going to express them in a little bit different way. So it could be floral color, it could be leaf color, if it's variegated. Sometimes the leaf shape might be a little bit different. The height, the growth habit and form of the plant. We have columnar varieties of trees, for example. We have dwarf varieties. These are varieties. So genetically, if we look at like a globe spruce, genetically it's the same as your typical Colorado blue spruce. It just is a different representation of the genetic makeup of the plant. So that would be, again, phenotype. And that leads us into species. So, again, species, varieties, all this talk, all members of a species share the same genotype or combinations of genes. So again, when we were talking about that, species versus variety, if you were to run, recombinant DNA analysis on a 50-foot tall blue spruce or a dwarf blue spruce, it's going to come back as blue spruce. Okay. You may see that different sets of genes have been turned on and turned off. However, genetically, you're not going to see any difference when you run an assay on those two plants because, again, they are the same species. So they usually do have same or similar phenotypical expression. We can think about that. Let's go back to the blue spruce again. They're still blue spruce. So they're still forming that same thick, waxy cuticle on the needle that gives them that inherent color that we've all come to appreciate. At the end of the day, they do share the same or very similar phenotypical expressions. So I mentioned him a moment ago. One of the most important people involved in this aspect is a gentleman named Carl Linnaeus. So a little bit of history trivia here for you. If you ever go to BW3s or Damon's again, if you play the Trivial Pursuit, maybe this will come up. And you can be a plethora of useless knowledge. So Carl Linnaeus worked to extensively catalog and track plant ID based on phenotype. So, we think back several hundred years ago, we didn't have the ability to do, genetic analysis. And so they really relied on the phenotype, the visual expression of these traits. And so that's how we got started in the taxonomic identification of plants. A little bit further on, in the 1800s, a gentleman named Gregor Mendel that you may have heard of if you've had biology before. He's a Catholic friar. He was obsessed with peas. I don't know why, but he was obsessed with peas, and he studied in his pea fetish. He studied the changes in phenotype of the seedlings from cross-pollinating different plants. So, I guess he was one of the first people that really started to dive into pollination. And really, if we think about it, he kind of, what he was doing really significantly increased our understanding of plant reproduction. And, this is where we start to kind of figure out that there's something more to what's going on in the plant, there's some sort of something. They didn't know that it was genetics or DNA at this point, but they knew that there was something going on. And he studied the changes in the phenotype of seedlings from cross-pollinating. So if you had, say, a pink pea and a white pea, he would breed them. He would take the pollen from one and cross it to the other, and vice versa. And then he'd grow those plants out, let them produce the seed, the little peas in the pea pods, and then he would harvest the peas, and then he would replant them, and then he would wait and grow them out again and see what they looked like. And so he started to figure this out. And if you remember, if you ever had biology, you might have done an exercise in biology called the Punnett square. And he really developed all of this. So this was, he started to actually figure out the probability that one trait would be expressed over another. And so this is really the foundation of our understanding. Gregor Mendel is really, I guess you could call him the father, no pun intended, but the father of modern genetics, because this is where we really started to understand something more was going on in the plant. And again, what he was actually doing would be what we would call breeding and hybridization. Okay, and as we move along in our turfgrass class, and if you've seen in other classes, when you go to a garden center or something, you see all of these different varieties of plants. Or we see all these different varieties, like in your seed exercise that you were doing, going and looking at the varieties in the seed bag. So this is actually a result of breeding and hybridization that was pioneered by Gregor Mendel. So again, considered the father of modern genetics. And you see here's a nice little picture of him where he's actually taking the pollen off the flowers and this is actually, pretty common today. I used to do a little bit of floral work when I was at Scotts too. We were working on EFL, extended flower life trials. So in the wintertime, when I wasn't necessarily doing a whole lot with the turfgrass trials outside, we were working with petunias, and we were actually trying to work with something called ethylene senescence. So senescence, if you might have talked about this, senescence is where, senescence is basically drop. Okay, senescence is where the flower drops, or in the fall time we have leaves fall in the fall. That is the process of senescence. And so extended flower life focused on the idea of kind of slowing that process down, as the name suggested, extended life by senescence. So I did a little bit of work with this. It's kind of interesting to watch because, we would actually go in and we'd have little, these little like clothespins, and, we would go in and we would count and we would mark different flowers on the plant. Most of what we did was more involved with flower counts to see if the trait was actually holding in the plant, if we had a viable specimen to send on for further genetic analysis. It was kind of interesting because it brings back memories of me working in the greenhouse back in the day. Again, going back to Linnaeus, we have the Linnaean domain, which, again, we created this whole structure back during this Renaissance period of how to identify living things. Obviously, the main focus at that time, ironically, was plants. Plants were the first things that led to taxonomic identification and classification of living organisms. Plants were very important. They're still important today. Many of the cures that we get for various diseases, many new treatments that we develop for cancer, and many things that we develop for other ailments in our own culture actually come from plants. You think about medicine doctors in the the rainforests, herbalists. We still have a lot of herbalists. We have a lot of people that practice homeopathic medicine. There's another Dr. Weil. I have no idea if he's related to us or not, but he is very big in holistic and naturopathic health. So plants actually are little pharmaceutical factories, and a lot of the compounds that they produce have very beneficial effect on human beings, just overall health. But there are plants that do produce certain, chemical compounds that are very beneficial. So it became very important during this period of time to help identify plants that were, as I call naughty, or plants that were nice. Some of them produce compounds that will kill you or make you very sick, but other ones produce compounds that are very helpful and beneficial. So plants really led to, kind of when we think about biology today, I mean, it really started in the plant kingdom. So as we work through it, and this is kind of the old school, the old school Linnaean domain, we get into eukaryotes, which are, they have organelles and they have membranes. So, if you think about prokaryotes, bacterium, the DNA is just kind of, everything's all kind of floating around in the cytoplasm inside. They do have their outer, shells, the protein shell and the membrane, but other than that, all the other organelles and everything, or everything that's going on, just kind of floating around inside the soup inside. So we have this diversification where we actually start adding structures inside of the cell. And, photosynthetic organisms were some of the first eukaryotes on Earth. So, your cyanobacteria and, different protozoa and stuff like that. Single-celled organisms, but they started actually creating these different, organelles, with their own membranes. The DNA actually ends up becoming confined inside of a nucleus instead of floating around loosely inside of the organism. And this really is what gives rise for plants to have the ability to photosynthesize, because again, you have to compartmentalize these actions inside the plant in order for them to occur. It can't just be happening in the soup, floating around in the soup of the cell. So, this ability to kind of have these organelles and membraned organelles and compartmentalizing processes within the cell is what allowed plants to exist. So the kingdom is the plants. The phylum would be angiosperms or flowering plants. The class that grasses fall into is monocots. Okay. So that means that they have one cotyledon in the seed, which we'll get into in a little bit here. You have an order, so you have the grasses and the sedges under the monocots. So if we back one up, monocots, there's a lot of different monocots. Onions are monocots, daylilies are monocots. So we have a lot of monocots besides just grass, okay? There's a lot of plants that are monocots. And then you get into the grasses and the sedges, and then you get into the actual family, which would be Poaceae, which is the grasses specifically. And then you get into your genus and your species. And your genus and your species is really genetically where you get into these, when we talk about the genotype and the phenotype, we really get down into the genus and the species at that point. So genetics is actually replacing phenotype as a more accurate determination of lineage. And you remember, back in the day, that's all we had, several hundred years ago. But as we have the modern era and the advent of molecular genetics, and it's getting easier and cheaper and faster to do this genetic analysis, we're now able to gain a better understanding of the lineage of different species. And some species are turning out, they're being renamed because they may resemble one plant, but they're more genetically similar to another. So you can have similar phenotypes in different species. It doesn't mean that they're related. You could have brown hair on a horse, you could have brown hair on a dog. It doesn't mean they're related. It's a similar phenotype. So, not that we were that ignorant when we were classifying things over the last several hundred years. It's just, it's a bad illustration of what I'm trying to say, that, genetics is just a much better all around, determination of the genetic lineages of different species, and whether or not they're actually species to begin with, or not just some, phenotypical variation of another species. So the APG4, the angiosperm phylogeny group, is what we currently use to classify plants. So it tries to marry the Linnaean system based on morphology, which morphology is the study of organisms and physical structures. Again, if you've had a bio class in your past, you might remember that. So morphology would be the study of the organism and their physical structures, which this what we're doing in this session actually is more of the morphology and biology aspect of grass in general. So under the APG4 system, families are much broader, but they're more inclusive. So we're kind of shrinking down the number of families. We're recognizing that, we didn't have all of these, families, or we didn't need all of these families because so many of these things are genetically similar. So families are much broader and more inclusive. And we see this introduction of subfamilies and clades, okay? Subfamily or clade. You use them interchangeably. A clade, if you ever see the word clade mentioned, it's referring to this subfamily class. So again, it's we have these subfamilies or clades because we're still kind of gaining an understanding or a knowledge of how these things are related. We're still trying to determine, if they are their own distinct pathway or if they just kind of level up to one of the other families. And so that's kind of where we know that these things are similar. They share a same pathway at some point historically, but they're still different enough that we don't quite know what to do with them yet. So we come up with this new thing called a clade. So monocots, again, we go back to monocot. What is a monocot? It's a seed that has one cotyledon. What the heck is a cotyledon? It's an embryonic leaf. Okay, so every seed of every plant has an embryonic leaf in that seed, so that as soon as it pops out, it grows that up, it uses the nutrient reserves in that seed, and it elongates that petiole from the seed, which ends up turning into a stem. And at the end of that is the cotyledon, which is the first embryonic leaf. Interestingly, a lot of times the embryonic leaf doesn't look anything like what the mature leaves are that the plant ultimately produces. So in the monocot side, one example I can think of is crabgrass. When that sucker starts to germinate, it almost looks like little tulip leaves coming up, but don't be fooled because it turns into something much more heinous after that. I mean, it's called crabgrass for a reason. So, looks cute and adorable when it starts coming up, but that's the best it's ever going to look. So dicots have two embryonic leaves. So monocot 1, dicot 2. There's your Latin terminology, mono and di. Great. This plays a role in herbicide selectivity. So if you don't remember anything else about monocot versus dicot, just know this. When we get into the herbicide discussion and when you move onward and upward with your careers, whether a plant is a monocot or a dicot will play a role in herbicide selectivity. We have monocot and dicot selective herbicides. So that, especially in turfgrass, becomes very important when we think about herbicides. When we're trying to control broadleaf or dicot weeds in a monocot turfgrass stand, I think you can see where a dicot selective herbicide can be very important. So these new subfamilies that we talked about, we've again, genetics has helped clarify a genetic lineage, and as you can see, so we have 3 subfamilies here. The 2 that we focus on for grasses would be the BOP. It used to also be called the BEP, and the Pac-Mad. Those are kind of cool. It makes me think of Pac-Man, the old game, but Pac-Mad clade, and the BOP clade. So basically, the BOP clade, you have your bamboos, you have rice, and you have your cool season grasses, okay? And then your Pac-Mads, is kind of everything else. If there's, I don't know if we really need to get into the specifics of the BOP versus the PAC-MAD. Again, the panacoidy, the aristoidy, and the chloridoidy. These are all different genuses of grasses. So these are the PAC-MAD clade. These would be part of the subfamilies that house a different genus and species of grasses. The important thing to know between these two, and we're going to get into this in a minute, is the PAC-MAD gives rise to your C4 grasses, and the BOP clade gives rise to your C3 grasses. So what the heck is a C3 and a C4 grass? Okay, well, C4, these would be, we also call them our warm season grasses. So if we think about warm tropical areas on Earth, these would be our warm season grasses. And then these would be our cool season grasses. So anything that's growing in our northern and southern temperate regions. So your BOP clade and your PAC-MAD clades give rise to your C3 and C4 grasses. This is an important distinction. And this is something that is really disrupting the traditional Linnaean model of taxonomy because, again, relatively new concept. I mean, not super new, but we knew there was a difference. We didn't know why. We didn't understand it. We're starting to learn a little bit more. Again, if we think back to some of the climatic changes, going back to Module 1, where we were talking about the advent of the ice ages, not every place on Earth was cold, okay? There were parts of Earth around the equatorial regions that were very hot, and they also happened to be very dry. And so we believe that those climatic conditions is what gave rise to the C4 traits that we see in grasses and some other plant species. Now, only about 5% of plant species on Earth are C4, carbon 4. The rest are C3. But it's an interesting, I guess if you want to call it or think of it as a mutation, it's an interesting mutation or, adaptation that occurred. It's not extensive, but it does increase the plant's ability to manage water. So again, if we think about hot, dry conditions, it also gives plants the ability to continue to photosynthesize in extremely warm temperatures, which is important because for these processes to occur in the plant, that you need a kind of, there's an optimal range. Plants are cold-blooded, just like a cold-blooded animal. They don't have the ability to regulate temperature like we do. So their temperature is from the environment. And depending on what the temperature is in that environment, they're going to do very poorly or very well. If we think about wintertime or hot, dry summer, do we see grasses in our lawns growing? But we know that there's a period of time in the spring and a period of time in the fall where they grow really well. And that gets back to, again, that there's this optimal temperature window for these plants to kind of work at peak performance. Okay, so they're going to grow really well and they're going to do what they're supposed to do really well. And outside of that window, it just drops off. Think of like a bell curve. There's this optimal peak of temperature and climatic condition and it drops off. So the C4 actually, it kind of moves that bell curve a little bit further up the range where cool season plants will not tolerate it. Okay, if you try and take a blue spruce and plant it in Florida, it will die. It cannot survive in those temperatures, okay? It normally grows on cold mountaintops in the West and primarily Colorado, hence the name Colorado blue spruce. So it's a higher elevation plant and it likes colder temperatures. Surprisingly, they do remarkably well around Northeast Ohio. Looking outside, I guess I know why. Okay. But we are probably, if you think about that species, for example, we probably are kind of the limit for a blue spruce. You know, I think if you try to really get much outside of Ohio, go further south, unless you're at a high altitude where it stays cooler, probably not going to do well. And these grasses are just the same. If you try and take, you know, cool season grass like Kentucky bluegrass and grow it in Florida, it's not going to do well. Now, we have made some advances moving these into more of what we call the transition zone, which would be kind of like Kentucky, Tennessee, northern Georgia. If we think about a band running across, you know, over from the mid-Atlantic over to, you know, like Oklahoma, northern Texas, things like that. You know, and then the Carolinas, this would be considered the transition zone. And some grasses do really well in it. Warm season grasses will not grow there. It's too cold for them. But some of the cool season grasses, we have actually hybridized them to perform reasonably well in those types of regions. So again, C3 and C4 are carbon 3 and carbon 4 metabolic pathways. Okay. I'm trying to boil down some of the science. My goal here is to just kind of give you an appreciation or an understanding of what's going on. If you know, you want to learn more about it, great. If you want to go down the rabbit hole like I did at one point in my life, that's awesome. But just understand, I'm just trying to give you some appreciation for what's going on in these plants. And you can see in the image behind here, there's something different going on between these two plants. So all plants have C3. Okay, so I'm going to confuse you more here. So all plants have the Calvin cycle. Okay, the 3-carbon Calvin cycle. C3 is what 95% of plants on Earth use exclusively to convert water and carbon dioxide into glucose. All right. So, you know, and if you know plants, the difference between plants and animals is plants have the ability to do a couple of interesting things. One is they produce their own carbohydrate. All right. So plants can't run and hunt and gather food and so on and so forth. So they've kind of come up with a neat mechanism in that they produce their own carbohydrates. And then they also do this other really cool thing, which is they produce their own amino acids. So we don't have the ability to do that. We have to eat it. If we don't, we die. Plants have this remarkable ability to do it on their own. So one of the processes that plants use to produce their own carbohydrate is, or the process that they use rather, is the Calvin cycle. It's also called the citric acid cycle. So this is what makes a plant a C3 or 3-carbon plant. Okay, and again, if we look here, we're bringing carbon dioxide in. We have this huge enzyme called rubisco. And then we have phospholipids that basically get involved, and boom, we're making ATP, which, if you remember anything from biology, ATP is your energy currency inside of the plant. So this is how the plant actually stores energy. It actually stores, we do this too, we produce ATP, okay, and ATP cycles in and out, okay? So we see it's cycling in and out of this process, becoming either adenosine diphosphate or adenosine triphosphate. So it basically, this is the metabolic process where the plant stores energy and then gives that energy back. It uses quite a bit of energy to produce carbohydrate, and that's where this carbon cycle comes into the plant. It's actually taking carbon dioxide from the air and turning it into glucose, okay? And then it is generating ATP at the same time. And that's how it's creating an energy reserve in the form of carbohydrate, and it's creating an energy currency molecule in the form of ATP. So some examples of these three carbon grasses would be wheat, oats, and rice. Okay. So this would be, in that BOP clade, if you remember the Oryzidaceae family, that is your rices. So that's basically rice is, I think, the only member of that family at this point in time. It's kind of its own unique thing. So cool season grasses use the three carbon pathway. So in our landscape today, when we think about turf grass, this would be things like Kentucky bluegrass, perennial ryegrass, tall fescue, and creeping bentgrass. So these would be your cool season or three carbon grasses. These would be at least four of the primary ones up here. Okay, so C4. C4 is an adaptation that 5% of plants on Earth use in addition to the three carbon cycle. And what it allows them to do is, it's kind of a neat trick. It allows them to reuse water, and it also allows them to reuse some carbon dioxide, and it captures and reuses some of these where it normally would be given off as a waste product. It allows a plant to kind of capitalize on the CO2 and use it a little bit. It just increases that efficiency a little bit more. It also gives plants the ability to survive in extreme temperatures. So this would be temperatures above 90 degrees. And so it helps reduce water use, and it helps increase CO2 efficiency. And if we think about it, during the Ice Age, one of the things that we saw was a significant drop in CO2 levels. So when the CO2 levels were high, that was all well and good for your three carbon plants. But what happens when your CO2 levels really start dropping off? What happens when they drop down below 300, below 200 parts per million? All of a sudden, these three carbon plants, they can't work as good, okay? And remember, they're passively absorbing this carbon dioxide from the atmosphere. So if a plant develops the ability to do a little bit more with the CO2 that it has available, it's going to live, okay? And that's where we saw the rise of these plants, because honestly, if most of Earth was covered by a ball of ice, the only place left really for a lot of these plants to grow and survive was in a lot of these hotter regions, these equatorial regions. And again, if most of the water on Earth at that time was frozen and on the surface, I mean, most of the fresh water, we didn't have atmospheric moisture levels at this period of time during the Ice Age. So, there was a lot of, there were dry spells, pretty significant dry spells in these arid regions, these arid equatorial regions, that they weren't getting rain. It was frozen out of the air in the form of huge glaciers on the northern and southern hemisphere. So, and in fact, Antarctica and Greenland are two examples of remnants of this Ice Age. That is actually, those are both what's left, from the Ice Age. And that's why we get concerned about whether or not we're going to push ourselves out of the Ice Age, because what's going to happen when all that water melts? I don't think Antarctica is going anywhere anytime soon, but there is an awful lot of ice up on Greenland. And that probably could raise ocean levels, a few feet. So not a dire situation, but definitely, going to have an impact if all that water melts off. So, again, the four carbon cycle allows plants to make glucose more efficiently with less water and respiration. So they don't have as much CO2 to work with. They don't have as much water to work with. They got to hold what they have inside of them. And it's interesting because when you look at cacti and succulents, they actually have this other cycle that goes on with them. I don't want to mislead you. They're actually there's a whole nother thing with these guys called the CAM pathway. So we're not going to go down that rabbit hole. It doesn't really pertain to turfgrass, but I just threw this up more for illustrative purposes that, when we look at plants growing, think of the desert, right? Or think of the Mediterranean where we have these hot, arid conditions and these plants have developed the ability. C4 is an important part of succulents, but they have something a little bit different going on in them as well. Again, just kind of illustrating, what kind of region do we see, carbon, 4-carbon plants growing in. And again, this would be a representation of one of these arid, hot climates. So corn is a C4 grass. We don't always think about it. We see it a lot up here in the summertime. But there's a reason why it only grows up here in the summertime. There's a reason why we plant it in late spring, as close to the drop dead window as we can for crops, which is usually, I think, around the middle of June. So that's kind of the goal, is to get it in when we have those warm temperatures, as close to the cutoff date for the crop insurance as we can get. It is a 4-carbon grass. Another grass here pictured is common Bermuda grass. This is a turf grass. It's actually grown pretty extensively in the South. And this is again, this would be another 4-carbon grass. Some of our weedy grasses like crabgrass are C4 plants. So they can survive in seed form up in our area, but they are a 4-carbon grass. So, one of the hallmarks of your 4-carbon weedy grasses is that they take hold and actively grow when the C3 grasses aren't. So if you think about in our landscapes here in Northeast Ohio, you've got a perennial rye or a blue rye lawn, or a tall fescue lawn. Those lawns aren't actively growing, or they're not growing at a very fast rate, usually over the summertime, especially if we're in 80 and 90 degree weather and there really isn't a lot of rain. No, they're not doing a whole lot. They're just kind of hunkered down, sitting there. You might see some dormancy setting in. They do lose a little bit in the warmer weather and the warmer soil temps. There is some death that occurs in the grass over the duration of the summer. But again, if we think about it, if that grass is just sitting there and it's not actively growing and it starts thinning out, it's creating opportunities for this guy to get going. And crabgrass, there's a few different species of crabgrass, but crabgrass, we treat it the same, we deal with it the same. It's not as important as knowing what to do when we get it. And this guy won't start germinating up here until about middle of May. So again, warm temperatures. So 55 degree soil temperature or above consistently. It needs some consistent airtime temperatures day and night. Needs adequate moisture to get going. So it really capitalizes on that warm, humid weather that we have here in the spring, going into early summer before things start to get hotter and drier. And again, it's actively growing when the other cool season grasses aren't, which is part of what allows it to cause us so much headache in the landscape. So it thrives during hotter weather and dry conditions. So this would be a perfect picture of like a kind of a low maintained lawn, probably typical of a lot of lawns or grassy areas that we see in kind of like an unmaintained or low maintenance setting. And you can see where the cool season blues and ryes are thinning out. they're checking out, they're going dormant, and look at all this bright green, like neon green stuff all over. It looks happy as a lark, you know. One good place to see something like this, honestly, is drive through a cemetery in the summertime. And you'll just see, I mean, it'll be like fluorescent green when you drive through there. They don't do a whole lot, unfortunately, to maintain a lot of the cemeteries. And so that's a really good place to kind of see some of this stuff going on. And again, it just illustrates the point I'm making that, you get into early to midsummer, and this is what a lot of unmaintained turf areas in Northeast Ohio can look like. So there's a little bit different, what do I want to say? So morphology. Let's get into the morphology here between C3 and C4. So there is a little bit different morphology going on between these two subgroups of grasses. And so it's referred to as Krantz anatomy. Now, the Krantz anatomy is where all the magic happens. It isn't the magic. The magic would be that 4-carbon cycle that we've talked about in addition to the 3-carbon cycle. The Krantz anatomy is actually the structural adaptation or the morphology that differentiates the carbon 4 plant from the carbon 3 plants, okay? So some of this Krantz anatomy or some of the anatomical differences between these plants is the 4-carbon plants have tighter veins and they conduct photosynthesis in 2 cell types, the bundle sheath, which the bundle sheath would be these purple guys right here, and then the mesophyll, which is this, we refer to it, there's the palisade mesophyll and the spongy mesophyll. If you ever looked at a cross-section of a leaf, in biology, it's really cool. You can actually see any leaf will work. I mean, you could take like a maple leaf off a tree and dissect it and look at the cross-section, but you can actually see, the cross-section pretty much looks like this. The difference, again, is that you have tighter veins, so the vein diameter is smaller in the 4-carbon plant. So this would just be the vascular bundle in a 3-carbon plant. So again, the vein, it's got a bundle around it, a bundle of cells, just like the veins and arteries in our body do. Something's got to keep the blood and other material in the veins and arteries. Plants are the same way. So you have these bundle sheath cells or the vascular bundle cells, and then I'm sorry, I misspoke there. So the vascular bundle in the 3-carbon plant, the 4-carbon plant has these bundle sheath cells, and the difference is, again, they have the ability to photosynthesize in this, and then before the CO2 and stuff moves out of the plant and the water moves out of the plant, we have this additional mechanism that it's passing through just to give it a little bit of a boost. So 3-carbon or C3 plants only produce photosynthesis in these types of cells. So you have the blue ones, which are the spongy mesophyll, and then you have the palisade mesophyll. And interestingly, if we look at the 4-carbon plants, they don't really have this spongy mesophyll. Part of that, again, is they really don't have the luxury of losing water. The 3-carbon plants are kind of wasteful. They're used to it. It's like they have Applebee's every day. So they don't need to worry about stuff like that. But, your 4-carbon plants, they're kind of like your keto plants. So they're pretty frequently in starvation mode. Again, that would be some of the morphological differences between the 3-carbon and the 4-carbon plant. Again, improved nitrogen use and efficiency over C3s, less water loss, and they survive with lower atmospheric CO2 levels. So it's a slight improvement in efficiency that gives them the ability to survive pretty well in these hot, arid regions across Earth. So again, they have improved water usage over C3 plants. So if we think about a lot of our landscapes down in, say, the desert Southwest, Southern California, Arizona, New Mexico, Texas, these actually, the four carbon grasses, warm season grasses, if we can grow them there, they're much better at living in these arid, dry conditions. And, yeah, we still need to irrigate it to maintain them, but we don't need as much irrigation necessarily to maintain them as we would a cool season grass. A cool season grass growing in a place like Arizona, Phoenix, Arizona, for example, I mean, it's toast. It's not going to make it. So heat and drought tolerance in hot, arid climates. So that's one benefit that we see in our warm season C4 grasses for turf. So for these, so what caused this adaptation for these grasses? They first arose about 24 to 35 million years ago. So if we think back to the timeline in Module 1, our our grasses in general, the grasses in general started showing up on Earth somewhere between 55 and 70 million years ago based on fossil record. They occurred primarily in if you remember Pangea. They occurred primarily in the region between like the middle of Africa, or the middle of the African continent and the middle of the South American continent. So, this area must not have been as hot and as dry as it probably is today. And so if you think about the ice ages coming in, they didn't occur right around the time that the dinosaurs died, and they didn't occur right around the time that these grasses started showing up. And again, this is something that occurred in more than just grasses. So it was grasses, and it was some other, call it broadleaf species or coniferous species that also developed this adaptation. And again, something climatically happened on Earth, and I believe it correlates with the ice age that gave rise to this. So, something clearly changed. Much hotter, much more sunlight probably. And then so we can kind of see what we think is the evolutionary pathway that led to the C4. I hate when it does that. It's a C4 pathway, and you can see here that C3 photosynthesis adapted in cooler conditions, lower light, temperate environments. So probably a lot of overcast conditions. Maybe wasn't, if we're looking at temperate regions, you're not going to have the day length to portions of the year. So the first morphological change that we see is the development of the Krantz anatomy. And then we see the photorespiratory CO2 pump, which is this trait that the C4 grasses use, that they actually have the ability to kind of like pump the CO2 into these bundle sheath cells, which the C3 plants don't. So it's a little bit more, it requires a bit more energy on the plant's part, but the gain is significant relative to the expense. And, you may remember from biology, any adaptation that something has an expense, okay? When primates evolved in those early grasslands and became bipedal, that had an expense, okay? There was an energy expense that resulted. And ultimately, the goal of the natural realm is to maintain entropy, okay? So an entropy would be the state of order. It's this, energy conservation in the universe. And so entropy and homeostasis kind of go along with each other. And so for an adaptation to occur, that means that some energy is going to be spent at some point in the process, okay? And for human beings, we grew upright, we became bipedal, we became much faster, okay? So we were consuming more energy in that regard, running around in these savannas in Africa and stuff. The other thing that happened is our brains grew. You know, and you think about that, I mean, that's part of the reason that our gestation is so long in the womb or in utero, is because, you know, we have this brain that's really complicated. And, you know, our brain consumes, at least on average, about 20% of the total energy expenditure of the body is the brain. You know, so if you think about a normal resting state, you know, we're 2,200 calories per day, roughly. About 20% of that is our brains. Okay. So there's the energy cost, right? But is there a benefit? And does the benefit of having this big brain outweigh the additional energy that that brain has to consume? Well, I like to think so. You know, I think that there's a benefit to it. But, you know, biologically, yes, there is. You know, biologically, our brain's ability to store information and kind of problem solve and create things like tools, okay? All of these things, all of this ingenuity that comes out of our big brain allowed these early humans to survive on the savannah. And, you know, so it is an important adaptation if you think about the survival of the species. And at the end of the day, that's what it's all about, guys. It's about the survival of the species. And so plants are no different. They're going to evolve. They're going to adapt. They're going to do things. It's going to help them. It's going to cost them a little bit. So it's a short-term loss for a long-term gain. Okay, and so this is kind of what we think the changes in genetic expression and enzyme activity that kind of led to this this adaptation that allowed for C4 photosynthesis. So again, when we think about latitude, location, and effects on temperature, you know, we look at Earth and you know, we don't always think about this, but it's a good thing to talk about at this point. You have your mid-latitude and you have your low-latitude regions on Earth. Okay, so low-latitude would be closer to the equator. Mid-latitude would be in these temperate regions. So North America, Canada, for example, mostly lie in this mid-latitude, as does most of South America. And Europe also lies in the mid-latitude region. And you have places like, you know, your rainforests in northern South America and Central America, and then also in Africa, like the Congo region. You know, you have these warm tropical regions, but then just on either side of these, they're kind of bordering or at the northern limits of these low latitude region on Earth. That's where we see these hot arid zones, and, you know, deserts. So you think about the Sahara, the Mojave, you know, these deserts often occur, you know, kind of in this area that's straddling the mid latitude and the low latitude regions on Earth. So about 30 degrees north and south longitude is really kind of where we see these arid regions. And I think that, you know, that kind of gives you some insight. So right around where we see the Tropic of Cancer and the Tropic of Capricorn is where I believe we see the rise of the C4 plants. So combination of heat, drought, and salinity. Again, this is a good representation. So, you know, on Earth, you know, we look through here in these green regions of South America and Africa, and also over here in Indonesia. Okay, so this would be kind of, these would be the tropical regions, your equatorial low latitude regions on Earth. So right around the equator, which would be 0 degrees longitude. The interesting thing about this is, we think about, well, why wouldn't we have a lot of C4 plants there? Well, because that region stays pretty stable. I mean, when you think about things like Hawaii or Indonesia or even the rainforests, I mean, where are our temperatures? Our temperatures typically stay right in that, call it 75 to 85 degree range. Okay, so it's very, very habitable for 3 carbon plants because, let's think about it here. If 5% of plants on Earth have this 4 carbon adaptation, why? I mean, and then if we think about it, you may have heard this before that some of the most ecological diversity, especially with plants, occurs in the rainforest regions. Okay, so think about those two things. If we have all this diversity, but only 5% of plants have that trait, that means they're not probably coming from these tropical regions. Okay. Now, I'm not saying that you can take a tropical plant and plant it in Ohio and it's going to survive. Okay. These plants have obviously adapted and evolved. For one, they can't handle the wind. They'll blow right over. Go down to Florida next time there's a hurricane and you'll see that play out. Okay. So there's not a lot of wind. There's not a lot of air circulation along the equator. And the temperatures actually stay relatively consistent, okay? Because there's this exchange of warmer air rising up from the equator, spinning up to or spinning down to the north-south pole, exchanging that heat, okay, cooling down, and then coming back down, warming up, and coming back to the equator. Okay, and this occurs as a result of the rotation of Earth. So, well, I guess what I'm saying is it's basically the Earth's a big heat exchanger. You have the warm air moving from the equator, cooling as it moves toward the poles and the northern latitudes and longitudes, and then it cycles back down around. Okay, so this area stays relatively constant. So, what areas on Earth are extremely sunny, extremely hot, extremely dry, have wild fluctuation? Well, here you go right here. So we have this border region here. We have another border region here down. These would be like where your African savannas are. And you look at these border regions all around Earth where we have these arid conditions, and you're going to have a lot of sun. You're not going to necessarily have as much air exchange going on. You will at points, but then in between, it's extremely hot. Okay, it's dry. So these are all things that contribute to the adaptation of these plants. Okay, so again, 5% of the plants on Earth have this adaptation. Where would that be occurring? Probably in these transition zones between our warm tropical regions and these hot desert areas on Earth. So, you have a combination during the Ice Age of the aridification and declining atmospheric CO2 levels due to oxygenation. All right. And if we look at CO2 historically, if we go back 600 million years, and we look at some of what's going on here, carbon dioxide levels were very, very high early on in Earth. And so you get to a period here, where we call this period here between 300 and 400 million years ago. There's this huge extinction that occurs, and, there's this event on Earth called the Great Oxygenation, and that's where a lot of this photosynthetic life starts getting involved. And you can see, the oxygen levels just crash, okay? Then clearly somewhere between 200 and 300 million years ago, there was some sort of cataclysmic event on Earth that caused another extinction and killed off a lot of life. So again, we have this rise in CO2 levels, and we kind of see this constant dance on Earth back and forth with oxygen levels on Earth as we move through this period of time. And, the big question is when we get out to this current day, this current era that we're in, again, if you look toward the end of this graph, you'll see much lower. This is where we're probably even below 300 parts per million, possibly even dipping down below 200 parts per million CO2, or getting close to that point. Obviously, we're sitting right now, we're probably about 400 or so, a little bit over 400 parts per million atmospheric CO2. So the question is, where are we going to go from there? I got a touchy mouse. I'm sorry, guys. So where are we going to go? We don't know. All right. We don't know if man-made CO2 emissions is going to spike us to the point that we can officially end the ice age. We don't know if Mother Nature is going to prevail and send us back into that ice age. Okay. But, I guess the takeaway for this is when we look at this period of time, okay, so again, 55 to 70 million years ago, so somewhere in here, so somewhere between the Cenomanian and the Paleocene era, somewhere in this era is where the grasses start forming. So right around the Paleocene, and maybe a little bit before, we see the grasses, okay, and then boom, here goes, we start going into decreasing CO2 levels. Hey, maybe grass played a role in those decreasing CO2 levels. It did become a very dominant feature on Earth's landscape during that time, and it is very good at doing what it does, okay? So, maybe it gave rise to the ice ages that we have today. I have no idea. But what I can tell you is we can see this decline in CO2 levels, okay, into our current era. And the adaptations that occurred between the C3 and the C4 grasses, those adaptations allowed many of those grass species, not a huge percentage, but allowed a percentage of those grass species to survive. And, when we look at the C5 plant makeup on Earth, a big chunk of it is grasses, okay? The Pac-Man clade, if you look at all of those grasses versus the BOP clade, so again, this adaptation occurred early on in the genetic evolution of these plants, okay? Grasses, CO2 levels were much higher than they are today when the grasses first started showing up. Okay. The BOP grasses were able to survive as the CO2 levels declined, but they became less and less, their ability to survive in those dropping CO2 levels was less and less. Okay. So as the CO2 levels fall, these plants can't survive as well. Something had to happen. CO2 is dropping, the climate's changing, and so some of these grasses develop the ability to survive in hotter, drier conditions and with lower CO2. And I think it occurred in those, I think what probably happened, I don't know, but my, creative brain's thinking that during this period of time, that region that we looked at in the low latitudes, was much drier and hotter than it is today. And so I think that when we see a lot of these grasses taking foothold on Earth, the cool season grasses had the ability to survive in the more temperate regions, like in North America, where a lot of the tree species died because of the cold and dry conditions. The grasses, the cool season grasses, had the ability to survive in those regions to a point. But then we have this other phenomenon going on with all these Pac-Man grasses. Those were in temperate regions, okay? And I think, again, something must have happened in these tropical regions where, they may have become more deforested through this period of time of the glaciers, and the grasses evolved and adapted to the conditions. So that is it for this one, and I will talk to you in the next module. And again, as always, if you guys have any questions, please don't hesitate to reach out to me. I'm more than happy to help you. Any questions you have, any landscaping questions, any questions about the class assignments, text me, email me. If you want to call me, you can. Just make sure you leave a message because my phone probably will block the call if it doesn't recognize the number that's coming in. So if there's nothing else, I guess we'll wrap it up here. And I will see you in the next module where we will actually start getting into the specific grasses. So that should be really fun. It's, hopefully something you guys have been waiting for is let's get down and break down all these grasses and the benefits and the differences, similarities, all that stuff. So, all right, I will see you guys next time. Thanks.